The goal of PNOCOOB is to test the clinical benefit of using molecular profiling to determine an individualized treatment plan in children and young adults with high-grade glioma. And the underlying rationale is using an individualized treatment approach based on whole exome and targeted panel as well as RNA sequencing. And the feasibility of therapy recommendations within 3 weeks has previously been demonstrated, of course, within the framework of PNOC-003, during which no major safety concerns with regards to the treatment plan being the individual treatment plan being designed within that time frame for up to 4 FDA approved drugs in combination was ever a concern. And the primary objectives of PNOC-008 are to determine progression-free and overall survival at 12 months in high-grade glioma cases that underwent safe surgical resection as well as focal radiation therapy. Additionally, secondary objectives are to determine the safety and describe the toxicity associated with the personalized treatment recommendations from the molecular tumor boards. But a challenge with respect to therapy recommendations within the framework of this trial is that therapy recommendations are limited when using only somatic DNA alterations, as like any pediatric CNS tumor, high-grade gliomas are low mutational burden tumors with a median TMB of less than 1 mutation per megabase. And to put this into further context, more than half of K27M high-grade gliomas, as you can see in the work done by many D3B investigators, do not have a potentially druggable target. And among those with druggable targets, the majority harbor a DNA alteration in a gene product involved in only a single canonical targetable pathway. So further presenting a challenge within the framework of combination therapies. So one potential solution to this is leveraging transcriptome data as an orthogonal strategy to further guide these treatment decisions. And the CBTN data within the context of PNOC-008 has been critical to help fully power this type of analysis. And the approach is generally to leverage the CBTN data to contextualize PNOC-008 patient of interest and identify nearest neighbors in mutational and transcriptomic space, and additionally characterize network and pathway level characteristics of nearest neighbors, and also integrate knowledge bases to prioritize targets and pathways on the basis of known drug-gene interactions and reference expression profiles of drug treatments. And this is all driven by patient to CBTN comparisons, as I'll illustrate in the coming slides. So this is just a simplified schematic of how we power our target identification using CBTN data, where PNOC-008 patient is compared to a background of either GTEx normal, all of CBTN irrespective of tissue of origin, and CBTN high-grade astrocytomas. For classical differential gene expression analysis, which then becomes the vector for being able to explore drug gene interactions based on well annotated knowledge bases that you see shown on the screen, to be able to make highlight FDA approved drugs that can directly target those differentially expressed genes. Additionally, there's more of a signature-focused, as you can see to the right, a more signature-focused analysis method that's driven by the development of the NIH links reference signatures, which I'll also discuss in greater detail. And going a little deeper within the transcriptome, CBTN data has been critical to the identification of high-grade and diffuse midline glioma transcriptomic clusters, from which we can characterize co-expression-based network modules with the underlying goal of being able to identify targetable network hubs, and with the underlying rationale as well that targeting these hubs will most greatly perturb the cancer network and be able to mitigate any mechanisms of acquired resistance to therapy. And part of this workflow as well is taking the CBTN patients, identifying the nearest neighbors to PNOC-008 and the PNOC-008 patient in transcriptomic space, And given the characterization of these clusters as well as these network modules, we can then relate these modules to pathway level characteristics to contextualize PNOC-008 into a biological setting. Additionally, immunophenotypic properties that may be shared with the PNOC-008 patient as well, in order to identify, again, consistency in biological state powered by RNA sequencing analyses. And then from these nearest neighbors, also be able to identify shared mutations that may also be driving the tumor. So this oncoprint is an illustration of a test case of a PNOC-008 patient whose tumor harbored an H3F3A K28M mutation. And you can see in the boxed region that there were a number of CBTN cases harboring this mutation that also shared mutations in TP53, as well as a potentially targetable alteration within PDGFRA, particularly amplifications in PDGFRA that were shared directly with this PNOC-008 patient, showing the DNA-driven method of informing potential therapies. But from these nearest neighbors, we also characterize the transcriptomic alterations, and this tabular output is demonstrative of the type of output that we get when we compare a PNOC-008 patient to CBTN, whether that's all histologies or whether that's exclusively high-grade gliomas. And from there, using our knowledge bases, we can then identify potentially blood-brain barrier penetrant drugs that had previously been FDA approved. And it's from contextualizing this patient within the greater CBTN landscape and building biological networks that we are able to annotate these transcriptomic alterations as well as being a network hub or not within the scale-free network topology. Again, with the idea of targeting these network hubs to fully perturb the most optimally perturb the cancer signaling network. And thinking with respect to this network biology and contextualization of the patient, the PNOC-008 patient's nearest neighbors, as well as their transcriptomic cluster assignment, are used to be able to relate the underlying network modules that are that comprise that signaling cluster, and through overrepresentation analysis, be able to compare or rather characterize the biological functions that are at play. And in this particular instance, this PNOC-008 patient was most similar to other high-grade gliomas that seem to have an active immune microenvironment characterized by the reactome pathways of interferon gamma signaling, cytokine signaling in the immune system, as well as the pathway for chemokine receptors at the top. And furthermore, in the bottom plot, another module characteristic of this transcriptomic cluster to which the PNOC-008 patient maps within the background of the CBTN data is that of extracellular matrix modification and cellular invasion. And so from this, we're able to derive a biological context as to what may be the most likely efficacious targets given the biology that's at play. And furthermore, we're able to leverage alternative tools such as immune cell enrichment analysis, looking at the background of CBTN data relative to the PNOC-008 patient of interest that's annotated in red in this particular plot. And from this, we can see, for instance, that this PNOC-008 patient is highly enriched in cells of the myeloid lineage, including M1 and M2 macrophages. And that highlights potential therapeutic strategies such as TAM re-education, the re-education of tumor-associated macrophages by simply targeting STAT3, which is a potentially viable target within pediatric high-grade gliomas. And furthermore, looking at other types of analyses that help to bolster the immune story even further, such as leveraging clinically relevant immune signatures and looking at the expression of that immune signature that's known to be associated with clinical response to PD-1 blockade from a prospective cohort analysis of a pan-cancer cohort. And you can see here in this particular case where the patient, PNOC-008 patient is designated by the dotted line, you can see when compared to the CBTN cohorts, particularly that of high-grade astrocytoma, a very high outlying expression of this immune signature, known as a tumor inflammation signature, would indicate that this patient is highly likely to respond to immune checkpoint blockade relative to other pediatric cancers. Again, further highlighting the utility of using CBTN data to highlight features that may have previously been latent, but by using this data now become much more salient. Additionally, the classical differential expression analysis relative to of the PNOC08 patient relative to the CBTN data also drives what's known as a signature searching analysis, where and this was made possible by the development of the Library of Network-Based Cellular Signature, or the LINCS consortium, that by the NIH, which put together a comprehensive database of reference drug profiles across at the level of tens of thousands of drugs tested on 70 cancer and non-cancer cell lines, to be able to create a database of signatures of drug response that one can then query. And in this case, by taking PNOC08 patients' differential expression profile relative to that of the CBTN, you can then look at the concordance between the expression, the ranked differentially expressed genes relative to known reference perturbation profiles. And another particularly useful element of this linked database is this touchstone data set that represents a subset of this data set, for which there are approximately 3,000 well-characterized compounds in terms of their mechanism of action, pathways affected, as well as target proteins. And so from this type of analysis, you can, from querying the ranked list of genes that comes from comparing the PNOC08 patient to CBTN subjects, you can then derive a ranked list of the most likely efficacious drugs, whether comparing to only high-grade gliomas or to all of CBTN. And what this enables is one to then start looking thematically at the type of drugs that are being recommended based on, or rather highlighted based on this signature searching methodology. And from there, you see that drugs that inhibit replication through multiple mechanisms, or those that are targeting the MAP kinase or PI3 kinase pathway for this particular patient, are coming up as reproducibly the most relevant pathways to target. But in terms of more comprehensively mining the types of pathways that are perturbed and that are most likely to be perturbed by drug treatment, one can take the full list of ranked drugs and perform a functional enrichment analysis, where, similar to gene set enrichment analysis, you instead have pathways described as lists of drugs based on the reference profiles from the LINCS database. And then from there, from one's ranked list of drugs that have been returned from the original signature search, you can then functionally enrich for pathways that are likely to be perturbed by drug treatment. And so this results in very rich visualizations, such as drug target networks that you see in this slide, where you can see the direct relationships between some of the top targets identified in a PNOC08 patient. Again, these targets being highlighted through a differential gene expression analysis against the CBTN data, and the potential drugs that can target these genes based on knowledge, again, from another knowledge base, the STITCH2 database, which consolidates both experimental and curated drug protein and protein-protein interactions, all with the underlying idea of using transcriptome data as an orthogonal method, and CBTN data really as the background to be able to identify what may be viable targets. So some of the conclusions from this, that CBTN data provides a wealth of molecular information to bring otherwise non-salient features to the forefront to inform potential therapies for PNOC08 tumor boards. And some of our plans are to potentially integrate multi-omic analyses, including that of CNS TAP, which will be discussed, I believe, in detail in the next talk, and other integrative analysis to be able to relate the mutational state to that of the biological signaling state in more meaningful ways. And lastly, leveraging data-driven approaches such as ML or network biology-driven approaches to guide our understanding of potentially synergistic combinations and limiting toxicities as part of this framework of transcriptome-driven drug recommendations. So I would like to acknowledge the team for a special thank you to Adam Resnick for his guidance and leadership. And additionally, the MIDI units at D3B who make all of this work possible, including members of the bioinformatics unit who drive our harmonized workflows to be able to generate the process data necessary to enable our scientific analyses downstream. Kamal Rathi, who generates clinical reports for PNOCOOB, and as well as the data, the adapters and clinical research units that also all help to make this possible. So that's all I have, and happy to take any questions.